All right, let's start looking at some of the individual components or structures that we can find within these cells. Now, remembering that we're looking at three key types of cells. We have our prokaryotic cells and our eukaryotic cells. The prokaryotic cells being the first cells, the simple cells, and we find these in bacteria and archaea. Remember that they belong to only unicellular organisms, and there are no membrane-bound organelles. Eukaryotic cells, on the other hand, developed later or evolved later in time. They evolved from prokaryotic cells. They can be both unicellular or multicellular, and they're going to be larger, more complex, and have membrane-bound organelles. Okay. Within those eukaryotic cells, there are two types of eukaryotic cells we're going to look at: the animal cells and the plant cells. As we go through this, keep in mind which structures you can find in all three cells, which structures you can only find in prokaryotic cells, which ones you only find in animal cells, and which one you only find in plant cells. All right, so the first structure I want to talk about is the plasma membrane. All three types of cells are going to have a plasma membrane. You can see that here. You can see the plasma membrane here in the plant cell, in the animal cell, and in the bacterial cell. Bacteria cell can have an additional cell wall as well as the plant cell. So your prokaryotes and your plant cells are going to have both plasma membranes and cell walls. The animal cells only have a plasma membrane covering it or surrounding it. And this makes sense because animal cells can travel to get away from disturbances and threats and things like that. And we'll talk a little bit more in detail about how the plasma membrane works. So again, the plasma membrane is your outer covering. It's what's going to enclose the cell and keep everything inside of the cell. It's also going to determine what can enter the cell and what can exit the cell. So basically, it regulates the flow of materials or transport of materials into and out of the cell. And this is really important. I'm not going to go into too far detail about this now. Just suffice to know that the plasma membrane, which is found in plant, animal, and bacterial cells, so in all cells, regulates the movement of materials in and out of the cell. In the next chapter, we're actually going to look at the plasma membrane in detail and how things move through the plasma membrane. The next structure I want to talk about is the cytoplasm, and this one is also found in all types of cells, and it's going to be kind of that interior portion of the cell. So where you find structures inside of the cell, so things like your ribosomes, your nucleate, or your nucleus. You're also going to have all of those encased within this kind of gelatinous liquid filled interior. Okay. For your eukaryotic cells, you're actually going to have a nucleus. And so this nucleus is a membrane-bound structure or organelle that is going to be found solely in eukaryotic cells. And you're going to find it in both the plant and animal cell. All the DNA will be housed within the nucleus. Okay. Inside the nucleus, you're also going to see this dark ball. This dark ball is not the DNA. This dark ball is actually what is known as your nucleolus, and it contains the material for making ribosomes. So what is called ribosomal RNA, which is what ribosomes are made out of. And if you remember, and we'll talk about it soon, ribosomes are going to make proteins. But in order to make ribosome, you need that RNA located there in the nucleolus. So within the nucleus, you're going to have that nucleolus, and then you're going to have a lot of DNA. And that DNA is going to contain the genetic material, which is organized in chromosomes. Okay? We're going to talk about that a whole lot more detail later in the semester when we look at genetics and how they're passed on. I should also mention that this nucleus, remember that it's a membrane-bound organelle, which means that the nucleus has its own plasma membrane that surrounds it. But in the case of the nucleus, it's a double membrane. So it actually has like two nuclear membranes that surround it to keep it very much protected and to really help regulate that flow of material into and out of the nucleus. The next structure we're going to look at are those ribosomes. So the ribosomes can be found in all three types of cells because the ribosomes are where proteins are made. And every cell needs to be able to have proteins in order to be able to do their functions and survive. So they all have to contain ribosomes. Ribosomes are also organelles, but they're not membrane-bound organelles. So they do not have a plasma membrane around them, which is why you can find them in your prokaryotic cells, as well as your eukaryotic cells. Okay. You can also find them in your eukaryotic cells specifically. You can find them either loose within the cell. Let me show you here. You can find it either loose within the cell, like you see some red little dots here with ribosomes, but you can also find it here attached to what is known as the endoplasmic reticulum. So your ribosomes can either be free-flowing, in the case of the eukaryotic cells, or it can be attached to that endoplasmic reticulum. So now let's talk about what the endoplasmic reticulum is. It's actually part of what we call an endomembrane system. And this is an interconnected system of membranes and organelles. I would highly suggest reading over this part in your book, which gives you a very good look at the endomembrane system and has some really great figures that encompass all parts of the endomembrane system. So your endomembrane system starts from the membrane, the type of plasma membrane that covers the nucleus, which is known as the nuclear envelope. And it continues on through. So the nuclear envelope will actually extend into what is called the endoplasmic reticulum. And there is a smooth endoplasmic reticulum and a rough endoplasmic reticulum. It then becomes the Golgi apparatus or complex, however you want to call it. It includes the vesicles, which are little kind of buds budding off. They're like little transport circles that are going to carry things between different places in the cell, or even exiting the cell or bringing things into the cell. These vesicles are actually made from the Golgi apparatus or the plasma membrane itself, where it just kind of pops off of those. Again, your book does a really great job of showing you some of this stuff. Lysosomes will also be found here as part of the endomembrane system. Remember that lysosomes are only found in animal cells. You will not find them in your plant cells. And then the plasma membrane itself. So the nuclear envelope extending all the way through to the plasma membrane forms your endomembrane system with all of these 6 parts involved in it. And this entire system is going to regulate the production, modification, and transport of proteins as well as lipids. And those include things like hormones and steroids. Your endoplasmic reticulum specifically, which is part of the endomembrane system, is going to be continuous with the nuclear envelope. So you can see here, this reddish here is the nuclear envelope, and then all the blue is going to be the endoplasmic reticulum. This side here has what looks like little bumps or measles on it, and those are ribosomes. So you can see here the rough endoplasmic reticulum is called rough because it has the bumpiness, has ribosomes attached to it. So you can imagine then that the rough endoplasmic reticulum is going to be in charge of synthesizing proteins. The smooth ER does not have any bumps on it, so it has no ribosomes attached to it. If it does not have ribosomes, it can't produce proteins. So therefore, your smooth endoplasmic reticulum is going to be in charge of making lipids. And lipids are your fats and your oils. If you remember from when you looked at macromolecules, your lipids can include fats, oils, phospholipids, and cholesterol, and different things like that. Okay. Your smooth ER, additional to making lipids, is also going to be in charge of metabolizing carbs. This means the breaking down of carbs. It's also going to be in charge of detoxifying drugs and poisons, and it's going to store calcium ions. So it's really important structure. So for instance, your liver, parts of your liver will contain a high amount of smooth endoplasmic reticulum because it can help detoxify things. So remember when we were looking at these cells and I told you, for instance, that this was an idealized animal cell? This means this is, in general, is what an animal cell can look like, but not all animal cells look like that. And I'll give you an example. In humans, our white blood cells kind of look like this, but our skin cells don't look like this at all. They actually look either square or rectangular, which is pretty interesting. And our nerve cells don't look like that at all. They actually look something like this. I'm a really bad drawer, but it kind of looks like fingertips and then like a little weird extension here and then more little fingertips, and then that's the nucleus there. or the cell body. It's kind of crazy. So you can see that different cells can actually look different ways. Not all animal cells are going to look exactly the same. Not all plant cells will look exactly the same. So this is what an idealized cell looks like. And they're not always going to have the same amount of structures within them either. So for instance, some cells might have a whole lot of mitochondria, which are these guys here for energy production. So a muscle cell will have a lot of mitochondria because it needs to do a lot of energy production. Whereas a liver cell might not have a lot of mitochondria, but it might have a lot of this smooth endoplasmic reticulum, because the smooth endoplasmic reticulum that doesn't have the bumpiness is going to be in charge of detoxifying, so removing things like poisons and alcohols and anything else that shouldn't be in the body, which your liver is in charge of doing because it helps filter your blood. Okay, so think of the clear functions of each of these structures. I would suggest taking notes if you're not already doing so of these different structures. You can expect to be quizzed about this in your midterm exam as well. The next structure I want to talk about is your Golgi apparatus or Golgi complex, or you can just call it Golgi. Okay, so the Golgi is a structure that's seen here and actually consists of like a bunch of flattened disc or membranous sacs. Okay, and what these sacs do is, as things are made in the smooth or rough endoplasmic reticulum, say a protein is made here in this endoplasmic reticulum, it can then move from there through a vesicle to the Golgi apparatus, where the Golgi will then do some final modifications on that protein and then package it up in another vesicle to then exit the cell to wherever it needs to go. So say you're making a protein like insulin, which is an important one, to then send to your body through your bloodstream to reduce the amount of sugar in your bloodstream. You would make it here in the rough endoplasmic reticulum, send it through the vesicle to the Golgi. It'll be modified, packaged into a new vesicle, and sent outside of the cell to enter your bloodstream. The Golgi, I should mention, also can receive things. So not just source and ships products from the endoplasmic reticulum, both rough and smooth. It can also receive products that are coming from the plasma membrane into the cell as well. Okay, the next one I want to talk about are your lysosomes, and this one is kind of my favorite. So the lysosomes are pretty wicked little guys. They're like little membranes, little vesicles almost. Okay, they're little round little guys. And what they do is they do something called phagocytosis. I love phagocytosis. This is where they will actually engulf smaller structures or substances to dissolve them and break them down. So they can help you with breaking down food molecules. They can also destroy old organelles or cells. And they can kill foreign pathogens. So say you got strep throat and your white blood cells are going to rush to your throat to help get rid of that bacteria, of that strep throat in your throat. Okay? So your white blood cells, are the cells in charge of killing it. But they're cells. They're animal cells. Therefore, they're going to contain a nucleus, an endoplasmic reticulum, an endomembrane system, vesicles, lysosomes, all of these different structures we're talking about. And they're going to contain a lot of lysosomes, like a lot of these guys, because they kill things. So these guys, these white blood cells, which contain a lot of lysosomes, will rush to your throat to help get rid of that pathogen to keep strep from overtaking your body and kill you. It's pretty awesome. If one of your cells itself gets worn out and kind of doesn't work right anymore, the lysosome within its own cell can destroy the cell and just dissolve it so that we get rid of old cells. It's pretty cool. Okay. And here is a really cool animation of phagocytosis. I highly suggest watching this one. It is pretty spooky cool. Okay, the next one I want to look at are your vacuoles. And these guys are pretty interesting. You can find small ones in some animal cells, not in all animal cells, just in some animal cells. Humans don't have any vacuoles, for instance. But some marine organisms are going to contain vacuoles, like contrail vacuoles to pump out excess water from the cells, or food vacuoles for helping to digest and break down food molecules. Central vacuoles are unique to plant cells only. So we're only going to find the central vacuoles in plant cells. And these are going to be huge vacuoles. They're going to be these really big round structures that can store water almost like a lake in the middle of the cell. And this helps maintain the shape and the size of plant cells. So generally plant cells can look like this, like a rectangle, and they can be connected one to the other to make a big plant. And each cell in the middle of it is going to have a large central vacuole that can fill with water to be able to maintain the shape of that cell. Okay? And that's known as turgor pressure. So if the cell has enough water, the lake will stay nice and full and the plant cell will stay with its beautiful shape that it has. However, if the cell does not have enough water, then what's going to happen is the lake is going to start to empty out, and then the plant cell will kind of start to droop. So if you've ever had plants in your house and say you forgot to water that house plant for a few days or a week or 2 or 3, right? And you forget to water it and all the leaves are droopy. They're just kind of hanging there. And that's because the plant doesn't have enough water. So these central vacuoles are basically running out of water and they're becoming very flaccid, making the entire cell kind of just bend out of shape. And that's why your plant kind of wilts. Once you put water back to the plant, if you haven't left it too badly that it hasn't died, what's going to happen is after a few hours, you're going to see those leaves pop right back up into shape. And that's because the central vacuoles will have refilled themselves. Okay. So to review this endomembrane system, we have the nuclear envelope here that's going to be continuous to the rough and smooth endoplasmic reticulum where we make lipids and proteins and also do detoxification. You're going to have vesicles that help transport things to and from places. We're going to have the Golgi apparatus or complex that is going to be able to modify and package these materials that are being produced. We also have lysosomes, which are going to help break down things. Okay. All right. We are close to finishing this PowerPoint, but we still have a few little things to go over. I am going to pause right here. And in the next video, we're going to start talking about the last structures. So again, we went over most of the structures in the interior of the cell, including those of the endomembrane system. Remember that the endomembrane system will only be found in eukaryotic cells because prokaryotic cells do not have membrane-bound organelles. So you only find this system within the eukaryotic cell, within those plant and animal cells.